Hi, I'm Kiran Musunuru. I'm an assistant professor of stem cell and regenerative biology at Harvard University in Cambridge, Massachusetts, and I work on the genetics of cardiovascular and metabolic disease. I'm here at ATVB 2013 in Orlando, Florida. It's May 1st, and this morning I gave a talk on our work entitled From Genotype to Phenotype to Therapeutics at Novel Myocardial Infarction Loci. And so in brief, what this work involves is trying to understand how changes in the human genome, how variations that occur between different people result in changes in cardiovascular traits, things like blood pressure and blood cholesterol levels and so forth, and then how that plays into cardiovascular disease, having heart attacks and other diseases of that type. And taking that information, understanding how the genes turn into disease presentations, and seeing if that can be used to give us insights that might lead us to develop new drugs, new therapies that could help people who are suffering from these diseases. So I told the story, or several stories, of several genes that we have discovered by using human genetics, by studying different people from different populations, in some cases very large groups of people, more than 100,000 people, and trying to understand what causes some people in a population to have disease and some people to be protected from disease. And in other cases, taking a few individuals who have very unusual disease presentations, have particular mutations that are so strong that they cause the patient to have a disease. So working with either populations and trying to understand common factors that many people share that lead to disease or contribute to disease, and then those few rare individuals who have very dramatic changes at the genetic level that cause them to have very unusual diseases. And so by understanding and identifying these genes, we're able to better understand the biological factors that actually lead to the disease process. And this all involves various factors at the genetic level, so the genes themselves, at the RNA level, so when genes are transcribed, they make RNAs, and then those RNAs are translated into proteins. Those proteins themselves have a variety of functions and act on other proteins and other chemicals in cells and in the bloodstream. And so the upshot is that we can work out how things translate, if you will, from the very fundamental level of the genes to what's happening in the human body. And so I told the story of three of these genes. One gene is known as SORT1, another gene is called AKT2, the third gene is called TRIB1, and these are all relatively new genes whose functions we don't understand that well, and so we're undertaking a variety of studies in different model systems, in some case mouse models, more recently in models based on human cells, and in particular human pluripotent stem cells, also known as embryonic stem cells or induced pluripotent stem cells, and using those model systems to better understand how genotype connects to phenotype, how genes lead to disease. And then the ultimate hope is that as we better understand how these things connect together, how genes lead to disease, we can find ways, find chemicals or find novel approaches by which we can intervene in that process. It might be as simple as a pill you take by mouth. It might be something more complicated like antibody-based drugs or even more sophisticated therapies, for example, cellular therapies. But the hope is that as we understand the process by which genes cause disease better, we can work on crafting therapies that then will eventually help patients both in preventing and treating diseases like myocardial infarction. Thank you so much.